For thousands of years, this deep red berry was trusted long before its mechanisms were ever measured. In an era before antiseptics, it was prized for stopping decay before it could take hold. Cranberries don't overwhelm the body like some antibiotics can. They work with precision, disrupting how microbes attach, how blood vessels respond, and how the gut ecosystem functions. That targeted strategy is what makes cranberries different from every other berry we eat. Cranberry, Vaccinium macrocarpon, is a fruit-bearing shrub with origins deeply rooted in the wetlands of North America, where it has grown wild for thousands of years. Unlike most cultivated fruits, cranberries thrive in sandy bogs and marshlands. These low-lying bogs are intentionally flooded at the end of the growing season, allowing the buoyant berries to float to the surface for harvest, a method that is both long-standing and practical. Long before modern agriculture, cranberries were used extensively by indigenous peoples, including the Algonquin, Wampanoag, and Lenape. They were valued not just as food, but also as medicine, dye, and a means of preservation. Crushed cranberries were applied directly to wounds, brewed into teas, and combined with rendered fat and dried meat to create pemmican, a highly stable, nutrient-dense survival food that could last through harsh winters. When European settlers arrived in North America, they quickly adopted cranberry practices from indigenous communities and initially referred to it as craneberry, inspired by the pink flower's resemblance to the head and beak of a crane. By the 17th and 18th centuries, cranberries were carried aboard ships and used on long sea voyages, prized for their resistance to spoilage and their ability to help prevent illness when fresh foods were scarce. Early records describe their use in preserving meats and treating wounds in an era long before refrigeration or antiseptics. As cranberry cultivation expanded in the 19th century, particularly in the northeastern United States, growers refined the bog-based farming methods still used today. Yet despite their growing popularity, it wasn't until the late 20th century that scientific research began to uncover what made cranberries biologically distinct. Researchers identified rare polyphenols capable of interfering with microbial attachment, offering a mechanistic explanation for centuries of traditional use. Today, cranberries are studied as a biologically active food, confirming what earlier cultures understood through experience rather than measurement. They exert their effects through a focused set of bioactive compounds that directly correspond to their health benefits. These compounds include A-type proanthocyanidins, rare polyphenols unique to cranberries that prevent bacterial adhesion to epithelial surfaces; anthocyanins, pigments responsible for cranberries' deep red color, involved in vascular signaling and oxidative stress regulation; flavonols, compounds that support endothelial function and metabolic signaling; phenolic acids and other polyphenols, compounds that interact with gut microbes and help regulate inflammation. Together, these compounds work synergistically, shaping microbial behavior, vascular responsiveness, and metabolic pathways. 1. Urinary tract defense through anti-adhesion. Cranberries' most well-established benefit is their ability to reduce the risk of urinary tract infections, but not by killing bacteria. Instead, cranberries prevent harmful microbes from attaching to the urinary tract lining in the first place. A comprehensive review concluded that cranberry products reduce the risk of UTIs in several populations, particularly women with recurrent infections. At the molecular level, this effect is driven by A-type proanthocyanidins, or PACs. A landmark study showed that these compounds inhibit the adhesion of pathogenic E. coli to urinary epithelial cells, effectively preventing colonization without promoting antibiotic resistance. Human studies further show that cranberry consumption produces measurable anti-adhesion activity in urine, with effects persisting hours after intake, reinforcing that this is a functional, real-world mechanism. This unique strategy, disarming pathogens rather than destroying them, sets cranberries apart from both antibiotics and most functional foods. 2. Vascular function and blood pressure regulation. Beyond the urinary tract, cranberries exert powerful effects on the vascular system. Clinical research shows they improve endothelial function, a key marker of vascular health. In a double-blind, randomized controlled trial, daily cranberry consumption significantly improved flow-mediated dilation, a measure of endothelial responsiveness in healthy adults. These benefits also extend to individuals with cardiovascular disease. A study in patients with coronary artery disease found that cranberry juice consumption improved vascular function and reduced markers associated with arterial stiffness. In adults with elevated blood pressure, cranberry supplementation has also been shown to favorably influence cardiometabolic risk factors, including blood pressure-related outcomes. Mechanistically, cranberry polyphenols enhance nitric oxide bioavailability, reduce oxidative stress within the endothelium, and support healthier vascular tone, effects that collectively contribute to long-term cardiovascular resilience. 3. Gut microbiome modulation. Cranberries don't just act on human cells, they also reshape the gut ecosystem itself. One study demonstrated that short-term cranberry extract supplementation significantly altered gut microbial composition, increasing beneficial bacteria such as bifidobacterium within just days. Rather than acting as probiotics, cranberry polyphenols function as microbiota-accessible compounds, selectively nourishing beneficial species while discouraging opportunistic microbes. This microbial shift may help explain cranberries' broader anti-inflammatory and metabolic effects, as gut bacteria play a central role in immune signaling and nutrient metabolism. 4. Suppression of H. pylori colonization. Cranberries' anti-adhesion effects extend beyond the urinary tract, even into the stomach. Helicobacter pylori is a bacterium associated with chronic gastritis and gastric ulcers. Clinical research shows that regular cranberry consumption can significantly reduce H. pylori colonization in infected individuals. One study found that daily cranberry intake suppressed H. pylori infection, reducing bacterial load without antibiotics. Earlier randomized trials also demonstrated similar effects, supporting the idea that cranberry compounds interfere with bacterial attachment to the gastric lining. This reinforces a recurring theme in cranberry research, altering microbial behavior rather than eradicating microbes outright. 5. Liver and metabolic health in NAFLD. Emerging clinical evidence suggests cranberries may also support liver health, particularly in individuals with non-alcoholic fatty liver disease, or NAFLD. Randomized clinical trials show that cranberry supplementation can improve liver enzyme profiles and cardiometabolic risk markers in patients with liver disease. A separate double-blind placebo-controlled trial further demonstrated improvements in metabolic parameters following cranberry extract supplementation. These benefits are thought to arise from reduced oxidative stress and inflammatory signaling within liver tissue, pathways central to metabolic dysfunction. Now that you understand how cranberries work, let's see some ways to choose, prepare, and use them day to day. Choose whole cranberries. Fresh or frozen cranberries can be cooked, baked, or blended into dishes, allowing you to benefit from their natural tartness and bioactive compounds. Cranberry juice. Many commercial cranberry drinks are cocktails with little actual cranberry. When choosing juice, opt for 100% unsweetened cranberry juice with no added sugars or other fruit juices. As a snack, dried cranberries can be a convenient option, but choose unsweetened or lightly sweetened varieties whenever possible. Pair with meals. Cranberries can be added to salads, grain bowls, or roasted vegetables for both flavor and functional benefit. Cranberry sauces and compotes. Gently simmer whole cranberries into a simple sauce and lightly sweeten if needed. Supplements and powdered extracts. Supplement capsules are an easy way to get a daily dose, and standardized cranberry powders can be blended into smoothies for extra polyphenol intake. In a world focused on powerful interventions, cranberries offer a quieter lesson. Their impact doesn't come from overwhelming the body, but from shaping how systems behave, how bacteria attach, how blood vessels respond, and how the gut ecosystem adapts. What began as a survival food in wetlands has become a case study in precision nutrition, showing how small, targeted biological interactions can create outsized effects. As nutrition science advances, one pattern keeps resurfacing. The most reliable tools for health are rarely new. They're familiar, accessible, and often overlooked. Progress doesn't always mean reinventing wellness. Sometimes it means understanding what's been part of our lives all along. So let's keep uncovering what else is hiding in plain sight. After all, health is wealth, and every bite is an investment in yourself.